My name is Thomas Schoenbeck. I'm a member of the math department here at Florida Atlantic, and it is my pleasure and honor to introduce Dr. Monica Hurdal, who is our Frontiers in Science speaker today. Dr. Hurdal is actually a native Canadian who got her Ph.D. in Australia at the University of Queensland in Brisbane. And her research was working on brain modeling, computing, and brain modeling. And she has worked on that since then, mostly changing a little bit the area of work. She has, after her PhD, she got a postdoc at the University, Florida State University. And then she had a short stint at Johns Hopkins University, and eventually she got a faculty position at Florida State, where she is now a professor of biomath and an associate chair of academic affairs. And her work is, in a way, could be described similar to the work for the brain, what Mercator did for the Earth in the 16th century, I think. So it's trying to map completely the brain, have it all mapped out so that anybody then can look at it and know exactly everything about it and travel around the brain the way we travel now around the world. Anyway, without any more, I should say that she has been a frequent speaker at conferences. She has been named Woman of Distinction in Science of Tallahassee in 2017. And she has a long list of publications in the area, has worked with a number of people, as a number of PhD students who's continuing their work. And I think I've said, without further ado, I should give the floor to Dr. Hurdal and let her speak. There will be a question and answer period after this. And I guess that's it.
Well, thank you very, very much. It's my pleasure to be here. I think a lecture series such as this is a fantastic opportunity for the community, and I think it's so wonderful to see so many people here. So today I'm going to talk to you about my research that involves mapping the brain, and in particular I'm going to talk a little bit about in health, aging, and in disease. So I'm going to give you a little bit of background. So the human brain, I'm sure you've all seen many pictures of the human brain. It's highly folded with valleys, and I'm probably going to use the word sulcus, that's a valley, a single valley, or sulci plural, or sometimes called fissures of the brain, and ridges, which are called a gyrus or gyri for plural. And what's very interesting about the human brain is the cortical folding patterns. Your folding patterns of your brain are unique, like a fingerprint. So everybody's folding pattern of their brain is unique. The global structure of the folding patterns is similar across everybody, just like everybody has two eyes, a nose, and a mouth. The local structure of the folding patterns are unique. So just like everybody's eyes have a different shape, everybody's nose is a different shape, the folding patterns of the brain are a different shape. Some might jog left, one might go deeper in somebody else's brain. So there's this high variability of folding patterns in the brain. Folding patterns in humans begin around the 16th week of gestational age and continue until after birth. Most of the folding patterns are completed by the time a baby is born, but there is still some folding that continues to develop after birth. So one of the things that I'm very interested in is, can we characterize the shape of the brain and the brain folds? So for example, if you have two individuals and say they're both healthy, their brains are going to look different in terms of their folding patterns. But now let's say you have two individuals and one brain is healthy and the other brain has some sort of disease or disorder, whatever you think that might be. It could be a mental health disorder, it could be schizophrenia, it could be Parkinson's, it could be Alzheimer's, whatever it is. But now we have this natural variability that occurs in brains. Can we determine what constitutes normal variability versus abnormal variability? So when you look at these two brains, is there some way that you can detect which one might have the disease and which one is healthy, because it falls within the range of what we call normal folding? So those are some of the questions that I'm interested in. The brain is so highly folded, we can't just look at it and decide what's going on. I mean, in some severe cases, there are disease where it's very obvious there's something that's wrong. But on the most part, if you look at a brain that has Alzheimer's, it's very difficult to tell which brain is a diseased brain versus the healthy brain. So this is where the mathematics comes in. And this is what I do. So can we somehow use mathematics to characterize the shape of the brain and brain folds? And how do these folds change in development? So from a fetal development, or as we age, just as we get wrinkles in our face as we age, the brain folds widen and deepen as we age. That's a well-known fact. And, how do they change in disease? So because not everybody's born with Alzheimer's, but Alzheimer's is a progressive disease later on in life. So my research interests, so I work at the intersection of mathematics and neuroscience. So in an area called biomathematics, so this might be a field of mathematics that you're haven't heard of or not familiar with. So this field of biomathematics, or mathematical biology as it's sometimes called, is where mathematicians use mathematics to work on problems in medicine and biology. It can be anything from brain data like what I'm looking at, to physiological structures, to diseases in biology or areas in biology like plant biology, or how fluids flow and things like that, or how blood flows, all different ranges of areas. So I'm interested in questions like what influences the size, location, and formation of cortical folds during development, because interestingly, there's no consensus in the neurobiology from neurobiologists as to how the folds develop in our brain, how they develop, where they develop, and why they develop. In fact, there's a number of competing different theories from the neurobiology point of view as to why folding patterns may develop in the brain. Of course, one idea is that, well, it's purely we want to constrain the surface area to volume. That's why we have brain folds. But it's not quite so simple as that. They have been able to do experiments in other countries and that where they've looked at how the brains develop in utero of, say, animals like sheep, and they've discovered that the folding patterns don't change. They've done experiments where they've been able to remove the skull. The folding patterns still remain the same, right? So it's not just a constrainment volume to surface area issue. So this is where I'm interested in how mathematics can contribute. So one area is mathematical modeling. So I'm interested in modeling the shape of the brain and how its folding patterns develop and grow over time. As I said, there's a number of competing theories from biology. So one areas of my research look at these different competing theories in biology, and I try to implement them mathematically to see what might be important in terms of these theories. For example, there's a biochemical theory that says, oh, well, at a certain point during development, there's an extra number of neurons that are created. Those neurons create high density, which in those high density regions creates folds in the brain, and in lower density regions, there's no folds. So that's one theory. Another theory is biomechanical, that certain axons want to maintain short connections in the brain for fast brain transmission, fast signal transmission. And so those axons want to draw the brain in together to make it during development to ensure there's short connections, short wiring, so to speak, and so that you have faster signal transmission. Another theory is what's called differential growth. The tissues in the brain develop at different rates. Maybe that development causes buckling as the brain grows and develops. So there's these competing theories, and of course, we can't go poking around people's brains, so maybe we can use mathematical modeling, right? We can create models that might look at these different theories, construct them from the science and the physics of it, create models, and see what happens. See, does domain, which means the shape of the brain, affect things? How fast it grows, does that affect things? Are there certain stages during development where maybe things are more critical that we can test with our mathematical models that we can't test with experiments? So that's where mathematical modeling can come in. So what I'm going to talk to you today about are some mathematical techniques for mapping the brain, because it's estimated that 60 to 70% of the brain is hidden from view when you take an MRI scan. So lots of stuff is buried in there that we can't see. So we need somewhere to start. So can we start with a map of the brain, right? Well, you might think that's pretty easy, but think of, something like that's highly folded, or even the curved, any curved surface, like a tangerine peel, an orange peel, right? How are you going to create a map on that? How are you going to unfold it? Well, to unfold it, right, if you've got a curved surface, you have to introduce tears in that surface, right, if you want to make it flat. Well, where do we want to do that, right? It's not quite so obvious and quite so simple. Well, it turns out that mathematics can help us do that. So the reason for doing this is to create these maps is to improve our understanding of cortical folding pattern formation in the brain. I'm trying to help understand what constitutes normal cortical folding variability, so we can maybe eventually use that to detect diseases. Who knows? And the other thing I'm interested in is identifying and discovering new mathematical metrics using what's called conformal, and I'm going to talk about that a little bit later, and topological invariance. So these are measures in mathematics that will stay the same if two things are the same, and they will change if two things differ, right? So maybe if two people's brain folds patterns are the same, they'll have the same invariant number, the same magic number that tells us how similar they are or how different they are from each other. So I'm going to give you a little bit of sort of biological background, because it's sort of important to, I think, understand sort of what's going on and what I'm going to be looking at with the brain. So this is a schematic view of the brain. So this is called the lateral view, or the outside of your head. And this is the medial view. So if you were to sort of cut down in the middle between your two hemispheres of the brain, this is what it would look like. So in this schematic view, this is the front and this is the back. So just some areas I might be interested in. This is the back of the brain called the occipital lobe. That's where vision is processed by the brain. This region here called the corpus callosum, that's what connects the two hemispheres of your brain and makes them talk to each other. We have this region here called the ventricle, which is a fluid-filled cavity. So we're all walking around with holes in our head that are filled with fluid that help cushion our brain. And this is very important in some of the other research that I do, because this is where neurogenesis occurs. So in early cortical development, as the brain develops, it's this structure of the brain where everything sort of begins and starts. So today I'm going to be talking to you a little bit about this area in the front of the brain called the frontal area, the frontal cortex. And it's actually sort of going to be right down here. It's called the prefrontal medial cortex. It's involved in affective disorders such as bipolar disorder and depression. And I'm also going to talk to you about this region of the brain down here called the cerebellum. The cerebellum connects to the brainstem, which connects to your spinal column, and the cerebellum is involved in things like hand-eye coordination. So as I mentioned, the human brain is highly folded. So we get a huge degree of folding across different species. So another application for looking at folding is that can give us insights into evolution and evolutionary patterns and trends across different species. So here we have some species that have little to no folding, such as this opossum. We have what we call medium folding, such as the cat or the lemur, and then of course humans, which have a high degree of folding. So you might ask, or what common question I get is, oh, well, do humans have the most highly folded brain? Do the number of folds you have, is that an indication of intelligence? Well, that's not true, and it's not so clear cut. And this is an example, right? This here is the brain of a dolphin. Dolphins are very intelligent. Are they more intelligent than humans? Maybe, maybe not. If you were to look at their brain folds, you might think so. But there are other species that have high folding, such as the species of zebra or a species of goat, right? So it's not clear what constitutes high degrees of folding and what the significance of it is. So with the research that I do, I mainly use anatomical data. So that comes from what we call magnetic resonance imaging data, or MRI data. I'm sure many of you in the audience have had an MRI scan of a knee or a joint, or maybe even of your brain. Typically, an MRI scan is taken in one of three slices. So hopefully you can sort of figure out, this is the axial slice, which sort of cuts through your head. We have the coronal slice, which would go this way, sort of up and down, and then the sagittal slice. So again, here you can see some structures. This is that corpus callosum that I talked about. But what I really want to point out here, which you can see clearly in this axial slice, is this light colored tissue and this dark colored tissue. The dark colored tissue is called your gray matter of your brain, and the lighter tissue is the white matter. So you can think of the white matter as your internet wiring. That's what sends the signals back and forth between the different areas of the brain. But the gray matter, which is about 3 to 5 millimeters thick in humans, is very, very important. That's where all your functional processing ultimately takes place. So the white matter transmits the signals, but the final processing occurs in the gray matter. And so because we're interested in the functional processing and where it occurs, neuroscientists are interested in looking at maps of the gray matter or maps of the surface of the brain. And they're interested in techniques for looking at ways of creating maps of this region of the brain, of the gray matter. So if we take those MRI slices and stack them one on top of each other, so if I take these slices here and stack them all up, because when you take a scan of your head, that's what you're sort of doing. So here, this white tissue here, that's your skull. The black is fluid, that's cerebrospinal fluid. And then, you have sort of a membrane and then the brain tissue here. So if we stack those slices on top of each other, you get something like this. And this is actually an MRI scan of myself that I had when I was a graduate student. I didn't have to shave my head. Hair is dead tissue, so it doesn't show up on an MRI scan. And what those dots are is they're actually oil capsules, cod liver oil capsules, because cod liver oil is high density and it shows up on an MRI scan. And so the reason I have this lovely expression is because at the time I was biting into like a mouth guard type thing where we could put these oil capsules on so we could sort of use them as a way of creating a coordinate system for the head and the data we were collecting. So this isn't very useful, right? So really what we want to do is we want to strip away the skull and the scalp and all the tissues we're not interested in and create reconstructions of the brain. So this is my brain. It's not really blue, or at least I'm told it's not really blue, right? Because it's gray, like gray matter. But a couple things. This is the front. This is the back. So here you can see blood vessels coming up on either side. This is the optic nerve that connects. You can see the two hemispheres of the brain, and you can see the high degree of folding. The other thing I wanted to point out is the brain is not perfectly symmetric like you see in those nice biology textbooks. And I'm not just saying that because this is my brain. Brains are actually not perfectly symmetric. And you can see that with this picture here. So this is two different brain scans. This is seen from the top. So I want to point out a couple things. So this one looks really rough. It's not that the brain is unhealthy or diseased, but when we take these MRI scans, depending upon the resolution that we take them at, right, they can look kind of, it's really a signal to noise issue for how we take the scan. So we do a lot of image processing on these scans to make them all nice and smooth and look pretty. That looks like this. However, this scan here is one of the best brain scans in the world. Even though it's about 20 years old now, it was done by a neuroimaging institute in Montreal. And this researcher, they scanned themselves 27 times, and they averaged the scans together to get one of the best signal-to-noise resolution scans available in the world. So you can see very clearly the different folding patterns of the brain. So I wanted to point out, right, so these are two healthy brains. So the scans are just taken sort of at different resolutions. But even though the folding patterns look very different, you can very quickly start to see some similarities when you start looking at them long enough. So for example, here we have sort of two folds and one that sort of jogs like this. Well, here you have two folds and one that jogs like that, right? So, but you can see that there is a high degree of variability in these two different scans. So I'm interested in, like I said, creating these flat maps of the brain. One of the reasons I mentioned is the functional processing is mainly on the cortical surface. So we want to do surface-based or 2D analysis-based methods. And this will lead us to this idea of creating cortical flat maps. Why is this a challenging problem? Well, what we call metric-based approaches, such as area or length preserving maps, will always have a huge amount of distortion. And there's no unique way of creating an area preserving map or a length preserving map from a surface that's highly convoluted, such as the brain. However, we can create another kind of map, which is called a conformal map in mathematics, or it's an angle preserving map, and they offer a number of useful advantages. The main one is it's mathematically unique. So if you want to try to preserve angles, no matter how you do it, no matter how I do it, there's only one way you can do it. So this gives us a very unique map that the mathematics can contribute. The other thing we can do is we can work in different geometries, and I'll talk about that in a moment. And then we can create, because our maps are unique, we can create unique or canonical coordinate systems. So advantages of brain maps, well, they allow us to compare different spatial relationships between different cortical regions. As I mentioned, we can create coordinate systems. But what this really allows us to do is compare individual differences in the cortical organization. We can try to identify and quantify specific regions where diseases may occur. We can analyze those regions that are buried within those folds. And even just as a visualization technique to look at maps of the brain. So what does conformal mean? So it means angle preserving. So basically, if you have two curves and a surface, and if you remember back to your calculus days and high school algebra days where you talked about tangents to curves, if you take the tangent to this curve and the tangent line to that curve, that angle between them is between those two curves is the angles that we're trying to preserve. Well, what does this look like? Well, if you have a rectangular grid, sort of like a square grid, it's sometimes called a Cartesian grid or just a rectangular grid, but just like graphing paper, and you apply a conformal mapping transformation to it, all the red and green lines still intersect at 90 degrees. So all the angles are still preserved. Now we have lots of length distortion and lots of area distortion, but we don't care. Our angles are preserved. So we know that how we navigate on the map will still be the same no matter which map we're sitting on. And this is really important. This is the map maker's problem, right? This is what led to sailors creating the longitude and latitude system, right? They didn't care about maps that distorted Greenland and made it look really big or really small. What they wanted is they cared about angles, how to get from point A to point B. And that's what we care about with our brain maps as well. So if we take a surface that's what we say embedded in 3D, so sitting in a 3D object, how can we possibly flatten that? So by flat, we mean a surface of constant curvature in mathematics. What does this mean? Well, we all know and love the Euclidean plane. That's a tabletop, a flat tabletop. That has curvature of 0 everywhere, right? There's no bends and no folds, it's just flat curvature. Another type of geometry we're all familiar with is the sphere. And that has a curvature of +1 everywhere. Well, there's another kind of geometry which you probably have never heard of called hyperbolic geometry. And it's related to what we call the unit disk or the circle. And it has a constant curvature of -1 everywhere. And I'm going to show you some examples. This is an area too where mathematics can bring in interesting ideas to fields that maybe have never thought of things before. So as I mentioned, why conformal? It's impossible to flatten a surface with intrinsic or built-in curvature without introducing metric or aerial distortions, but we can preserve angles. And this is like the map maker's problem that I mentioned. So the only theorem that I'm really going to show you today, because it's very important in a timeline, is called the Uniformisation Theorem. And it was created or developed and proved in the 1880s. And it's a generalization of another theorem from the 1850s. And in the end, I don't really care that you care about the theorem, but it is important from a mathematics point of view, right? This is very old mathematics that's 170 years old, that's being used in modern medicine and the research that I'm doing, right? This is why mathematics is important. And it's a continually evolving field, just like all the other sciences, right? A lot of people think, oh, you know, what is there to do in mathematics? We know everything about trigonometry already. We know everything about geometry, but that's not true. It's an evolving research field, just like every other science discipline. And so this is an example where very old mathematics is being used in modern research. So what this theorem says is there exists a unique conformal mapping up to automorphisms, which is a fancy word for transformations, from a surface to one of these surfaces of constant curvature, the Euclidean plane, curvature 0, hyperbolic disk, curvature -1, or the sphere, curvature +1. So what this theorem tells us is that if you have a highly curved surface, such as the brain, you can always find an angle-preserving map. It always exists. Now, this is what we call an existence theorem in mathematics. It only tells us it exists. It doesn't tell us how to find it. And that's where the real challenge comes in. So up until the 70s and 80s, it was really hard to compute a conformal map, an angle-preserving map, from an arbitrary object because it was just too hard. We knew these surfaces, these maps existed, but we had no way of computing them. Well, in the late 80s and early 90s, there were some mathematical developments, and of course, computation becoming really fast. There are now ways that we can calculate and compute conformal maps on highly folded surfaces, such as the brain. If any of you are engineers, you may have heard of conformal mapping in aerospace design and aerospace engineering, right? They're often used for wind foil and testing wind tunnels and wind flow design over airplane wings. Conformal mapping is also often used in video game design for texture mapping, how you make those nice fancy video games in fast and real time. That's all done with conformal mapping. So we know where conformal maps exist. So how do we create them? Well, when we create our MRI scan, and we look at it computationally, really what it's composed of is a whole bunch of little itty bitty little triangles. And that's what we call a triangulated mesh, right? So now if you think of these triangles, of a curved surface, so these little triangles all connected at various angles, and I'll show you some examples in a moment, right? If we look at the sum of the angle around a vertex, we call that the angle sum. So how can you possibly take a surface that's curved and make it flat? Well, what we're trying to do is we look at the proportion that each angle contributes to the overall angle sum, and that's what we want to preserve in our conformal map. So that's how we go from something that's highly curved to something that's flat. There are many methods now for computing conformal maps. A lot of them are numerical computational methods, but the method I'm going to talk to you about today is called a circle packing method. And it's really I think it's kind of cool because it's all a geometric method and it makes these really neat pictures, and I'm going to show you some of them. And the circle packing is a technique, so from these mathematical developments like I said, in the late 80s and early 90s, that if you compute what's called a circle packing, then you happen to get along with it a conformal map. So as a side effect, we get a conformal map. And it turns out a circle packing is really easy to compute. Well, what is a circle packing, I hear you ask? She's talking about circle packings. What does that mean? Well, simply put, a circle packing is a collection of circles with a specified pattern of tangencies. So all of our circles are tangent to each other. So here you can see we have all of these little triangles, and all of our circles are all tangent to each other. So again, I'm going to not really talk about the mathematics, but what the mathematics tells us is that if we compute a circle packing, then as I mentioned, we get a conformal map along with it. So how do we compute a circle packing? Well, we begin, so this is a curved surface, and this is just a rotated view of that surface. So we want to impose some very simple conditions. Basically, wherever we have a vertex of one of our triangles, we want to place a circle. And we want to require that whenever those two vertices form an edge, we want those circles to be tangent to each other. And then we also impose an orientation condition, so we're always going in the same direction, right? We don't want to flip any orientations around. So what our goal is to compute the collection of radii, right? So I'm just starting with circles. I don't know what radius they are. And I want to find the nice, perfect combination of circles, the radii of those circles, such that they all are tangent to each other when we have these conditions that two vertices must form, those circles must be tangent to each other. And that's called a circle packing. So the idea is really, really simple, and this is why it's so fast to implement. So let's begin with zero curvature, right? So if you have a triangulated surface and you have zero curvature, it's already flat, so you can find the circles such that they're all tangent to each other. It's pretty easy to do. You can even compute it geometrically. But now let's think of a case where we have positive curvature. So think of a paper cone, right? The only way to lay a paper cone flat is to cut it and lay it flat. But if you cut it and lay it flat, you have that gap, right? So that's because you have a deficit of angle. That angle sum, which needs to be 360 degrees to make something flat, is too small for a cone. So if we look at our circles, what we want to do is we want to decrease the radius of that center circle so that it gets smaller and everything becomes tangent to each other. Really simple idea. The other case is a saddle point. If you think of a saddle, right, we have too much curvature, right? We have more than 360 degrees. So if you were to lay that out, so think of like a lettuce leaf, right? That's all kind of ruffled. If you were to try to lay that out flat, the only way you're going to lay it out flat is you get an overlap with some of the big pieces of lettuce, right? So we have an overlap. So that's because we have too much angle. So what we want to do is we want to, in this case, increase the radius of the center circle to spread everything out to make everything tangent to each other. We apply that process repeatedly, and eventually you can compute the circle packing. And all it comes down to is the cosine law that you learned way back in high school. Who knew that could be involved in brain mapping, right? So what we're doing is we're taking the inverse of the cosine law, trying to find the angle. We use some geometry. We can look at our radii of circles, right? We have this circle here with the radii and this circle here. And we can construct some angles. And we want to sum all these angles all around, so they're 360 degrees. So the algorithm is a very simple algorithm. It's basically just adjusting all of the radii of these circles iteratively. And you might think, oh, well, if I adjust my circle here and make it too small here, that's going to affect something next to it, right? Might make it too big, might make a gap. But everything eventually ultimately filters out to the boundary. So, and you can prove computationally and mathematically that you can always compute the circle packing. So interestingly, the circle packing is easy to compute in the Euclidean plane. It's easy to compute in this other geometry called the hyperbolic plane, but there's no known algorithm on the sphere. So we have to employ a mathematical trick where we basically compute it in the hyperbolic plane and then can compute the mapping that way. But that's sort of an open question, is how do you compute these circle packings on a sphere? So here are some results. So here's the two hemispheres of the brain. So the curvature, the coloring is according to the mean curvature. So the bright yellow ridges are the ridges or the gyri of the brain, and the black is the valleys or the sulci of the brain. So here we have the lateral view from the outside and the medial view, and we can create this spherical map. And this is a sphere, so you could rotate it if you had it computationally. We could map it to the plane. So if you were to cut like a hole in your surface, so usually we cut it maybe where that ventricle area is that I talked about earlier, then we can map it to the plane. So this is our Euclidean map where we have zero curvature everywhere. And this is called our hyperbolic map where we have curvature of -1 everywhere. And the neat thing about the hyperbolic map is it's always going to be this disc-shaped, right? The Euclidean map, we use what's called a boundary condition on the edge, which comes from the geometric information from the brain. So its shape will always change. but the hyperbolic map will always be a disk, which is a nice way to look at things and compare things. So we can do something too. We can do a neat trick called using a little bit of computation and a little bit of mathematics and a little bit of video where we can create these cool visualizations. And I just have to exit out of here to show you my video. So we can create these maps where we go from the 3D surface and literally unfold and flatten our surface to create our flat map. Now our map, let me put it on repeat, it'll play. So even though it looks folded when it's flat, that's just using computer graphics and texturing for shading, but it is really flat. So this gives neuroscientists a really useful way to navigate from a flat map to the 3D embedded surface. So in the circles, right, we use the circle mapping, the circle packing we got to sort of map these circles on the 3D surface, just using like sort of a computer graphics, video gaming kind of tool to create this sort of texture mapping of what this looks like. Oh, back to the beginning. A few slides here. So we can create these maps of the brain. And so one of the things that my research group has been involved in is looking at other structures of the brain that maybe haven't been mapped before. So one of the structures is the cerebellum. So we detach it computationally from, excuse me, from the brainstem. And one of the things that's really interesting about the cerebellum is it's so highly folded, right? You can't even pick them up on all the MRI scan images, right? Those folds between the cerebellum are usually less than a millimeter thick. And so this is why not many people have looked at trying to create maps of the cerebellum. So what we did was we took our cerebellum, we had a neuroscientist label different structures on the cerebellum for us, and these are what are color-coded. And this orange corresponds to data from a PET scan, where they had subjects that had a little cylinder device, and they had to exert a force on it. And the force they had to exert had to match the force on a computer screen. So this is involving hand-eye coordination. You're seeing the force, you're exerting it on the cylinder and trying to match what the force is. Why were they interested in this? Well, this particular group I was collaborating with, they had a group of patients that had a hereditary disease called cerebellar ataxia, and this hereditary disease, basically part of the brain eventually just melted away and disappeared over time. And it was often misdiagnosed, right? So it involves hand-eye coordination. So they were using this and trying to study patients that had this disease. But this is where our hyperbolic maps become very interesting. So we have, many of you have probably seen a picture like this. This is done by the artist called Escher. And in this picture, this is drawn as though it's a hyperbolic map. And all of these angels have the same area hyperbolically. So, or the same area. So what does a hyperbolic map mean? Well, from the center of the map to the edge of the map is considered an infinite distance away. So what that means is things on the edge of the map look really, really tiny, even though they're all the same size. It's sort of like when you're driving. In the distance, things look really small, but then as you get up close, you see them for their real size. So one of the neat things with hyperbolic maps is we can do a very interesting kind of transformation called a Mobius transformation. So if you think of in the Euclidean plane, right, so in the world we all live, the only angle-preserving transformations are translation, rotation, and scaling. If you do anything else, you're changing the angle, right? Stretching it or anything like that. Well, in hyperbolic geometry, we have another really neat kind of transformation that allows us to pick on a point on the map, any point on the map, and move it to the center of the map. All the angles are preserved, and so this allows us to zoom in and focus on different regions of our map that might be of interest. And neuroscientists really think this idea is pretty cool. it's sort of like looking at things through a microscope, right? Things are in focus in the center, but a bit blurry on the edges, and you can move it around to focus in on whatever area you want to. So then we can start looking at comparing flat maps of different subjects, right? So here we have the cerebellum from two different subjects, our hyperbolic maps, and you zoom in, and you can quickly start to see sort of different anatomical differences on our flat maps, different functional differences based on these experimental results. So now we want to use mathematics to try to quantify some of these things now that we can create these maps. So there's another type of map we can create with these conformal maps that we're using to look at with twins. So I have some collaborators that have some data from twins. And they have both identical and fraternal twins. And what's interesting is, as with non-twins, identical twin brains have different folding patterns. Now, it's thought that they might be more similar than non-twins, just two siblings at random, but they are still different. And so they have some very interesting data with these twins where one twin is healthy and another twin might have a disease or affective disorder such as bipolar or depression. So can we use our flat maps to help identify similarities and differences in the curvature and our folding patterns of these twin brains? So we're looking at a region, they're particularly interested in this region called the ventral medial prefrontal cortex. This is a highly folded region. So this is sort of a tilted view of the brain, but essentially if you end up going straight up through your nose, this would be the region of the brain that you would be interested in, that you would hit. And so it's highly folded, so it's very difficult to study. This is that region extracted in 3D. So we were able to use our flat maps to create a map of a region and help them figure out which region that you specifically wanted to look at with this particular study. So we can then create coordinate systems. We can create our flat maps and then create coordinate systems, which can help the neuroscientists sort of navigate where they are. But one of the things that's really interesting is we can also create a rectangular map with our conformal maps. So as I mentioned with our Euclidean maps, if we use the geometry from our brain data, right, we get a map that's in the Euclidean plane, our flat map, but it always has a different shape. Well, we can specify what we call a different boundary condition. We can nominate 4 points on our conformal map so we can map it to a rectangle. And then it turns out that the aspect ratio of the width to the height of the rectangle is what we call a conformal invariant. It's sort of like this magic number that tells us, quantify sort of how conformal two things are, how similar they are in their conformality. So for example, you can never conformally map, so never do an angle preserving map from a square to a rectangle because they're just different objects. You can never get from one to the other while preserving angles. So this conformal invariant, this aspect ratio, helps us quantify this idea. So we can look at our data and we have, so our 3D surface, our Euclidean flat map, but now if we nominate like 1, 2, 3, 4 corners, we can map it to a rectangle. Excuse me, I'm getting over a cold from last week and I have a bit of a cough. So if we look at the aspect ratio from the width to the height of our rectangle, we can come up with this number, this conformal invariant. So now, if we have twin A, I mean, I don't know if it's statistically significant. This is what we're looking at. But boy, these two numbers are sure a lot more similar than the right hemispheres, right? So this maybe gives us a way of distinguishing left to right hemisphere and different regions of the brain that we might be interested in. So what are some of the other things we can do? Well, there's a whole host of things we can do with our conformal maps, right? So we can start with our 3D surface. We can cut out a region, map it to a rectangle. We can take our whole surface of the brain, map it to a sphere or a hyperbolic disc or a Euclidean plane. We can create our rectangular maps. But the other interesting thing we can do is if you look at these regions that are orange, we sort of simulated some functional regions on the brain. Well, we can find out a way, we can figure out a way with our circle packing to extract these regions that leave, if you say take out this one, it leaves like a hole because it's a circle. We can make it look like a circle in our map. And it turns out that the aspect ratio of the length to the width of the logarithm, sorry, not the length to the width, the logarithm of the inner radius to the outer radius is also a conformal invariant. So now we can start to extract all sorts of different regions, look at the radii of these different regions we're extracting. Now we have a whole set of numbers for a particular person, right? Once we have numbers, we can start doing statistics, mathematical comparisons, looking at different things. So maybe we can come up with a set of numbers for different people, different diseases, different regions of the brain to quantify how similar and how different they are. So where are conformal maps? Where have they been actually used? Well, they've been used in imaging studies of different structures of the brain, such as the hippocampus in Alzheimer's disease, looking at diseases like schizophrenia, the cerebellum, looking at hemispheric asymmetry. So this is a disease where maybe one hemisphere is a different size than the other hemisphere of the brain. There are other diseases where we have looked at called lissencephaly and microcephaly, where there's an excessive folding of the brain, abnormal folding of the brain, not enough folding of the brain, or the brain grows too small, right? This was big in the news from the Zika virus about 10 years ago. So these are all different things where conformal mapping and brain maps can contribute towards the study of our understanding of how diseases progress in the brain. And a slightly unrelated topic, but it's still very interesting. It turns out that what you see, so in our visual field, and how it's processed by your brain, where it's processed by your brain, is almost a perfect conformal map, right? They actually discovered this almost 100 years ago now, where a doctor was studying gunshot wounds from soldiers from World War I, and he discovered that this relationship between when they had wounds in the part of the brain that affected their vision, and what part of the brain the gunshot wound was and how their vision was affected, turned out to be almost a perfect conformal map. So where are we heading? Well, circle packings are mathematically unique. We can work in various geometries, such as Euclidean, the hyperbolic geometry, and spherical geometries. And they're very flexible in terms of mapping to different shapes. I showed you rectangles, but we could pick any geometric shape we wanted. Maybe it might be useful to map to a pentagon. So some of the future work that I'm involved in is comparing maps between different subjects. So this involves these conformal invariants and the metrics that we're talking about. We want to look at alignment of different regions. We want to align one volume of our surface to another, looking at the conformal maps and matching the conformal maps, how similar or how different. Sort of imagine taking one map and placing it on another map and seeing how similar or different they are. Can these conformal maps be used to characterize cortical folding, right? We can create these lists of numbers now. So how can we use those? And can mathematics be used to model cortical fold formation via reaction diffusion systems? So that's another area of research that I'm involved with. Where we're actually looking at how the brain develops over time that I mentioned earlier at the beginning of my talk. So I would just like to acknowledge, of course, this kind of work can't be done by one person alone. I've had many PhD students that have helped me, as well as many collaborators. If you want more information, you can always check out my web pages. I have some very cool, colorful pictures there. And thank you for your attention.
Yeah, I wonder if there are any questions.
studies using these with autism. Research looking at different functional.
Yeah. Yes. Yeah. 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 Maybe you should use the yeah.
So things are looking at the stage of more comparative, like do these techniques, can they actually be used to diagnose anything, right? So there have been a number of studies that have looked at Alzheimer's data to sort of see what is it similar and different between Alzheimer's and healthy individuals. So it's at this point, it's really more proof of concept. Another area where it's been applied is to demonstrate that in certain people who have schizophrenia, certain folds seem to be slightly different in schizophrenia than in healthy individuals. But you know that it's a difficult thing, right? Because you're sort of looking at subjects with a known diagnosis and making comparisons, right? Would you be able to arbitrarily detect that by picking somebody at random? It's hard to say. And of course, you don't want to misdiagnose. Yeah, but that is the goal. Yes. Yes. They're not like that, but in a lot of the research with regarding folding pattern development actually looks at ferrets, because interestingly, ferrets, their folding patterns almost develop completely post utero. So there's a lot of research looking at some of these biomechanical models and things like that in ferrets.
Yeah, I guess I don't know which. Let's go to the front. Gentleman here. Yes, you. Hi.
Right. So I guess one of the challenges with stroke is so this mapping, this brain mapping kind of work, this is purely a surface-based technique looking at the surface of the brain. So stroke, depending upon where the stroke happens, it could be looking at the interior of the brain. So in that case, that would not be, you wouldn't be able to use these techniques. But you could certainly use them to perhaps look at how the brain has changed after the stroke, right? So if part of the brain has changed anatomically due to the stroke because of blood buildup. I think one of the areas where these type of techniques could be very useful is in longitudinal studies, right? Eventually, we're going to get to the point, maybe, where we always have a brain scan every 5 years, right? So can you look at a trajectory of how your brain changes over those 5 years? And having techniques like this will be very important to help simplify the data so you can help sort of see what's happening with your brain trajectory over time. And I think that's sort of some interesting areas where this could be applied.
You had a question.
I don't really know. I'm not a medical doctor, so you know, I think it's hard to indicate. Right. Well, they are quite dynamic in the sense of they essentially erode over time, right? So as we age, our gray matter thins, our folds become wider, and they become deeper. But in terms of actual sort of structural changing after time, I mean, the brain continues sort of, the folding patterns, the primary folds sort of continue to develop until about age 3. The benchmark people sort of use in neuroscientific studies is the brain is developing, still developing up until about age 25. So then after that, is it starting to decline? At some point, it does, right? So there are different sort of theories about that. But in terms of after that point, you know, there's really no major structural changes other than the sort of erosion that occurs.
Is there a question?
Well, the better the MRI resolution, the more triangles you have in your image reconstruction, which means the more circles you have in your circle packing, which would mean the more accurate you get. So, so in some senses, no, there's not. It's more a limiting factor of the data. But what's always a trade-off with MRI images is what's called the field of view, right? Using an MRI machine, you can get like almost subneuron images of the human brain even if you are looking at like a half a square millimeter, right? But nobody wants to look at that. We want to look at the whole brain. So it's always sort of a trade-off between, you know, how much you can see. Just a regular laptop. Yeah. Yeah.
Yeah.
Yeah. Yeah. So the circle packing, especially in the hyperbolic and Euclidean maps, takes seconds.
I think we have.
Well, so Alzheimer's is an interesting disease. So Alzheimer's, the function is affected over time, right? You get different structural plaques happening. So sort of different neuroanatomical changes happening with the Alzheimer's disease. So Alzheimer's is a tricky thing to diagnose, right? Often symptoms for Alzheimer's are actually occurring way, way before you're diagnosed with Alzheimer's. They think as much as 10 years beforehand. So perhaps this could be used maybe again if we can understand how Alzheimer's might affect slight changes in the folds. Maybe this might be able to use to detect these changes a little sooner. Yeah. Dementia, so I think, yeah, that's a sort of a different category. It's not Alzheimer's, but it's sort of similar. I have not looked into any of that, but I think too, a lot of these things are subtle changes over time that are sort of really hard to look at, where you would need these longitudinal studies to be able to look at them in more detail. Yes. Yeah. Yeah. Right. So we take that into account a little bit by looking at different surfaces that we reconstruct. We can look at the gray matter cerebrospinal fluid surface, which will be different than the gray matter white matter surface. So when I first started working in this field, everybody looked at the gray matter cerebrospinal fluid interface, that boundary. But that turned out to be too hard because the brain folds were sort of too tight. That thickness made it too tight to create accurate surface reconstructions. So everybody now looks at the white matter, gray matter surface interface. But yes, if you were interested in what's going on in deeper structures, you would either need to use, just look at those structures separately, like the hippocampus, or you would need to use a different technique. It's sort of like the difference between, you know, mapping the surface of the Earth to look at the geography of the Earth. Well, that doesn't tell you anything about the interior of the Earth. Yeah. 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 There was another question. Yeah, I don't think anybody really knows that, but that's sort of a multi-million-dollar question, right? If you can figure that out, that would be extremely helpful, useful. Any other questions? All right, well, thank you very much for your attention.
Thank you for coming. I think Dr. Hurdal could stay here for people to ask her questions directly, but I guess we are closing this session now. Thank you very much for coming. They should let me do it.
Congratulations on your research.
Talk about multivariable.
Yeah,
We're using multiple variables. Regular correlation. Is essentially and that's what Einstein was using. Same curvature of space. Yes.
So what I think is, so I'm very interested